Okay, so picture this. Have you ever tried to read a book where you're forced to choose between, say, a blurry summary of the whole chapter
Mm-hmm.
or reading just a single crisp sentence, but the rest of the page just goes completely black?
That sounds like an impossible choice. It's the classic context versus resolution trade-off. You lose the forest for the trees.
Or the trees for the forest, exactly. And for a long time, that's been the struggle for scientists trying to read the regulatory code of our DNA.
Right, but that trade-off might finally be over.
And that's what we're digging into today. The new paper from Google DeepMind on AlphaGenome. Our mission is to understand how this model finally cracks that code without the sacrifice.
And cracking is really the right word for it. Before, you had separate tools for separate jobs. You had models like Informer or Borzoi that were great at seeing the big
The
picture.
long-range interaction.
Yeah, but the details are fuzzy. Then you had something like Splice AI, which was the opposite, super high resolution, letter-by-letter detail, but
Total tunnel vision. It couldn't see how a gene regulator thousands of letters away was affecting things.
exactly. Which brings us to the breakthrough. AlphaGenome processes a context of 1 million base pairs,
Which is that huge big picture view.
but it makes its predictions at single base pair resolution, so you get both.
And the performance is just off the charts. State-of-the-art in 25 out of 26 variant effect prediction tasks.
It also matches or beats the best external models in, what was it, 22 of 24 genome track evaluations. It's just a new benchmark.
You know, that idea of optimizing a complex workflow with AI, it actually brings us to our sponsor, Ambercilc.
Ah, a good transition.
Right. If you're looking at the power of AlphaGenome and wondering how to get that level of efficiency for your own business,
Whether it's automation or integration or software development.
you should really check them out. Ambercilc helps you uncover where AI agents can make the biggest impact for your work or even your personal life.
That's a great point.
So just head over to ambercilc.com for your AI needs.
Okay, so let's get under the hood. How does AlphaGenome actually pull this off?
It's that hybrid architecture, right? The UNet and the transformer.
Exactly. They have this unit-inspired backbone combined with a transformer tower.
So if I'm understanding this, the UNet part is like what you'd use for image editing to get that pixel-perfect local detail.
That's a perfect analogy. And the transformer part is what powers large language models. It's brilliant at understanding long-range relationships.
So it's treating the genome like both an image and a language at the same time.
Precisely. But the real engineering magic here is the efficiency, what they did with knowledge distillation.
The teacher-student model.
Yep. They trained massive teacher models, did the heavy lifting, and then taught a much smaller, streamlined student model to get the same results.
Which means researchers don't need a supercomputer to use it.
Not at all. You can run it on a standard NVIDIA H100 GPU and get an answer in less than a second.
That accessibility is just so important because we have to remember 98% of our DNA is this regulatory dark matter.
Right. It's not the genes themselves. It's the instruction manual for how and when to use the genes.
And if you can't read the manual, you can't fix the machine.
And the paper gives this incredible aha moment with leukemia, the tau-1 oncogene.
Yes, in T-cell acute lymphoblastic leukemia.
AlphaGenome figured out exactly how one tiny mutation created something called a neo-enhancer,
So it basically installed a new on switch that wasn't supposed to be there.
an on switch for a cancer gene. And because the model understands the 3D structure and all the surrounding marks, it saw the whole causal chain. Other tools just missed it.
And it's not just that, it's also predicting splicing, right? How RNA gets edited.
Yeah, there was an example with a variant in the tibial artery from the GTEx data that caused something called exon skipping, a critical error.
And AlphaGenome saw it coming, just from the raw DNA sequence.
It connects those tiny dots to these huge biological consequences. It's amazing.
And to top it all off, it's open science. The code is on GitHub, the model weights are on Kaggle and Hugging Face.
They're really putting the tool in people's hands. It moves us so much closer to understanding rare diseases and, true personalized medicine.
It's an incredibly optimistic time for genomics. It really is.
Absolutely.
It makes you wonder, if we can now read the regulatory code this clearly, how long until we can start comfortably, writing it to fix these kinds of errors? But that's a thought for another deep dive.
A big one.
If you enjoyed this podcast, please subscribe to the show. Hey, leave us a 5-star review if you can. It really does help get the word out. Thanks for tuning in.